Chapter 14 in our text is entitled The Fullness of God. This is an interesting aspect of God, or dimension of God, perspective on God. It's a fascinating verse. Jeremiah 23:24 says, I fill, God is speaking, I fill all of heaven and earth, says the Lord. I fill all the heaven and the earth. It isn't just that God is everywhere. He fills the universe. Christ, according to Ephesians 1:23, fills everything in every way. There's this concept that is a little bit challenging to wrap your brain around, or at least my brain around, that God is full, it fills everything, and He wants everything to be full. God fills the creation. He wishes to fill our mouths. He wishes to fill, in other words, with food. He wishes to fill the land with fruit. He wishes to give in abundance. God is abundant in His goodness, in His truth, in His forgiveness, in His grace, His mercy. He abounds. These are words that are trying to get this concept of what I call the fullness of God. He abounds in blessings and grace. He gives abundantly, and He offers a more abundant life. It's a beautiful verse in Malachi where, go ahead, give to me, God says, and see that I don't give it back to you, shaken down and then filled up again and overflowing. God gives richly. He is rich in mercy. He gives richly in mercy. He is perfect. He gives and He gives again. Repeatedly throughout Scripture, we get this concept that God isn't just everything and big, and He doesn't just have everything in abundance. It's beyond that. No matter how big you think He is, He's beyond that. No matter how much He gives more than that. He is generous beyond generosity. And that's what I'm trying to get the idea of in this concept of the fullness of God. God wants us to see that fullness, but of course it's like everything else with God, it's invisible. We can't see it. So God illustrates that goodness in various ways, and I believe He shows it, among other things, in creating organisms in abundance, an abundance of organisms, that diversity, biodiversity we spoke about before. We see this in Genesis chapter 1 with the creation of the sea creatures. Let the waters teem, which gives the impression of be full of sea creatures. And so He created them, and the waters did teem with things. And then, this is what's cool, He goes ahead immediately and says, now, be fruitful and multiply. So he puts an abundance of them in there, and then He tells them to keep on going, become more abundant, multiply from that point. There's the concept of the fullness. It isn't just biodiversity. It is filling things and filling things completely abundantly. So how does that work? How does God, in fact, illustrate that process of filling, of increasing your number? Well, we usually refer to this in biology as reproduction. So at present, in our present world, God illustrates His fullness, I believe, in the process of reproduction, which allows organisms and humans to fill things and to fill things abundantly. It's interesting that the way God has created reproduction always involves going through the stage of a single cell. No matter how big the organism in the process of reproducing, coming through, producing a new offspring, that offspring has to go through a single cell stage. Obviously, single cells start out as single cells, and they don't go to anything else. But the larger organisms that are made of multiple cells, God has chosen reproduction to be done in such a way as you have to start with a single cell and then build the number of cells from there. So even if you're a whale, the largest organism, largest animal on the planet, you are going to produce a single cell that then multiplies to become all the cells of the large whale. So for an organism to reproduce, the cell has to reproduce. So reproduction always goes through cellular reproduction. That's the only way that organisms can abound on this planet. So we're going to look at cellular reproduction. The reproduction of a cell is not what the cell does most of the time. There are several reasons for this. The primary reason is that during reproduction of a cell, when a cell is dividing to produce a second cell, it has to package its DNA in a form where it can't use it, it can't access it. And so it can't be in that stage very long, or it would do damage, in fact, kill the cell. So reproduction is actually something that occurs only for a short percentage of the life of a cell. Most of the life of a cell is not in reproduction itself, it is in non-reproduction. It's a normal stage of life. Once a cell begins the process of reproduction, there are several steps. The first step is to copy its DNA. Because a cell is going to become two cells, it wants its DNA to be copied in such a way that one copy can be given to the daughter cell, one copy can be kept by the original cell. So we have to copy our DNA. And this is called replication. DNA is untwisted, it's unzipped, copies are made of each side, and in the process, the center portion of the strand of DNA is left connected. So you unzip the DNA, but you don't completely unzip it. You leave it connected at one spot. So it creates something kind of looks like an X. And then you create the second strand of each of these, and they remain together, connected together. Now, the second thing that happens in the reproduction of a cell is everything outside the nucleus is divided. So let's say you have organelles that you have multiple examples of. You have multiple chloroplasts. Well, half of the chloroplasts will move to one end of the cell, half the chloroplasts will move to the other end of the cell, so that when the cell divides, you've got an equal number of chloroplasts in each side. Same with the mitochondria. Any of the cell components that you have more than one copy of, half of them go to one side of the cell, half to the other. But what about organelles where you've only got one organelle in the cell, such as the rough endoplasmic reticulum or the smooth endoplasmic reticulum? In that case, those particular organelles are split down the middle. They're just half of the organelle is just cut and sent to one side of the cell, half of it goes to the other side of the cell. It's a plasma membrane, it's fairly easy to do that, but that's all that's necessary to set things up so that the cell can reproduce. The third thing that must happen is the nucleus has to divide. This is arguably the most complicated part of the process, the trickiest part of the process. It's especially true that it's tricky to copy the DNA that's within the nucleus. And that there are two different methods that can be used by the cell to divide its nucleus, either mitosis or meiosis. There's two different ways to divide the nucleus depending on what you want in the final cell. If your purpose is to create two cells that are identical to each other and identical to the parent cell, to the original cell, then you use mitosis. Mitosis duplicates things exactly. So let's say you've got a cell that's in your finger, a skin cell in your finger, and you cut your finger so that a skin cell is actually damaged or destroyed. The skin cells next door to the dead cell detect the fact that there's a dead cell and say, ooh, we've got to replace our neighbor. So they are themselves skin cells, so what they need to do is produce exact copies of themselves to take the place of the cell that has been destroyed or damaged. Because they want to make exact copies of themselves, they use mitosis to divide the nucleus. So mitosis is used to repair damage. Also, if you want to, let's say, grow bigger, you need more cells. You're in the process of growing from a juvenile to an adult, and you need more skin cells. Again, if you've already got skin cells and you need more skin cells, you just need mitosis to create more skin cells. So mitosis is chosen by the cell if you want to repair or replace cells, or you want to increase the number of cells. But there's another option you might have if rather than produce exact copies, you really want to make the gametes or the sex cells. You want to make a sperm cell. You want to make an egg cell. That requires a particular cell to change, to be different from, it's going to start one way and it's going to end up different. It's going to end up as a sperm cell, as an egg cell. If you want to do that, if you want to produce what's called a sex cell or a gamete, you need to undergo meiosis in dividing the nucleus. The fourth thing that happens after you have copied your DNA, you have taken everything outside the nucleus and separated it into two parts of the cell. You have divided your nucleus and separated those things into two parts of the cell. The last thing that happens is the cell divides. It's called cytokinesis. Cyto means cell, kinesis is movement. It's actually cutting the cell in two. Now you're going to end up with two cells that are smaller than the original cell. The last stage is those two cells will grow up to the proper full size. So if you only had half the chloroplast in your cell, so now you've got to increase the number of chloroplasts up to an adult cell. You've got to increase the number of mitochondria up to the full adult cell size. You've got to increase the size of your endoplasmic reticulum, your Golgi bodies, and that sort of thing to restore everything so the cell is full-sized again. Now you can live normally as the cell and wait for the next time that you have to divide. That is the basic overview of cellular reproduction. The most challenging part of this is dividing the nucleus. So now we're going to look a little more closely at the dividing of the nucleus. First of all, we're going to look in this session at mitosis. If you want to make an exact copy of your cell, of your DNA, and put it into the daughter cell, you choose mitosis to do so. Mitosis involves several different steps in the process, and we give them names as phases. The first step, the first phase, is called prophase. Pro really means pre, it's the beginning phase of it. The first thing that we note in prophase is that the DNA it's already been duplicated, but we need to, what we call, condense it. Why are we doing this? What's going on here? Well, DNA in its normal form is an extremely long molecule. It's a big stranded molecule, and it's difficult in that form to move the DNA from one place to another. Reminds me of the experience when I was a kid. My mother used to use yarn a lot. She used to knit and things like that. We also had a cat. We had a cat that would love to get a hold of the yarn and carry the yarn all around the house and unravel the yarn from whatever form my mother had it in and take it through the chairs and under the tables, and there we go, we got a mess. Well, I was an obsessive compulsive kid, so I was just perfect for the child that my mother could say, "Kurt, why don't you get all that yarn back into a ball so that I can use it?" Because it was unusable, of course, stranded through all the furniture. In the process of doing that, I would just simply take the yarn and wind it up into a spherical shape. What I observed, which is interesting, is that when Mom would buy the yarn in the store, it would be in a form called a skein, kind of a hot dog shaped thing, where if you pulled the yarn at the exposed end, you could keep pulling for the entire length of the string, the yarn, and it would never get knotted. It would never stop. That I thought was pretty cool, because when I put the yarn into the balls, and if you tried to start pulling that end, it would get knotted up, and it never came out completely smoothly as it did in that skein. I always wondered, what is the special way in which you have folded this yarn to get it into a skein where you can get it out without creating knots? And of course, other examples of this, if you've got a hose and you're trying to put the hose back in a coiled form, next time you take out the hose, alas, it gets all knotted up. It's very difficult to lay the hose out in a proper manner that it doesn't get twisted when in fact you pull it out. Same with a rope. You're doing rappelling or something like that. It's a very special knack. You have to be trained in a special way to in fact lay the rope into a coil that doesn't get knotted when you uncoil it. DNA is, and when this yarn that I was putting into a ball, if I tried to take the yarn before I put it into a ball and try to move it from one place to another, because it was so long and twisted them around and in between things, I couldn't effectively move it from one side of the room to another. Or if I had two pieces of yarn that were wound around the furniture, I couldn't separate them in that form. What I'd have to do is put them into my ball of yarn first, and then I could easily separate them. I could easily move the yarn from one place to another. Same thing is true with DNA. In its extended form, in its usual form, where it can be copied, where copies can be made of it, it's in a long string form. That's great for making copies and getting copies of your genes, but it's not good for moving DNA from one end of the cell to the other. So if we're going to take our DNA and put half of it on one end of the cell and half of it on the other end of the cell, we've got to put it into what we call condensed form. We got to make a hot dog shape out of it in order to be able to then move it from one place to another. Also, it's got to be a very special shape. The spherical balls of yarn that I used to make weren't a good shape for unraveling the yarn after I've moved it from one place to another. It's the skein shape, a very special way of folding it that allows you then to put it back into its original form without it getting twisted. And the same thing is true of DNA. It's got special proteins stuck onto the DNA that allow the DNA to be folded into a hot dog shape that is very, very thick compared to its length, and it allows it to then be moved from one place in the cell to another, and then it can unravel itself without getting twisted. It's really kind of cool. When you're looking at this under a microscope as this is happening, the DNA normally is so thin in its extended form that you can't see it under a microscope. You will not be able to see it in lab, for example. However, once it goes into this skein form, once it's condensed, it actually becomes fat enough of it is folded on itself that you can see it. It appears under the microscope. And so when people were watching this, when biologists were watching this, they would watch the DNA appear. And these things that would appear actually responded to stains that you would put on it so that you could see the difference between things that are found in the cell. These bodies, somes, would appear, and they would appear to certain colors. So they called them chromosomes. These are colored bodies that would appear at one point in the reproduction of a cell and then disappear late in the reproduction of the cell. And so we called this appearance of DNA condensation of DNA. It's kind of like gas, water in the form of gas in the atmosphere can't be seen, but if it condenses into water droplets, we can see it. And so DNA seems to do that. Normally you can't see it, and then it seems to condense or appear in droplets hot dog-shaped things called chromosomes out of nowhere. We call that the condensation of DNA. So in prophase, DNA condenses. It appears in the microscope. Technically, at the point DNA first appears, that begins the process of meiosis. The second thing that happens in prophase is that the nuclear membrane around the nucleus disappears. We've got to be able to separate the DNA into two parts of the cell. We can't do that if there's still a nuclear membrane around it. So the nuclear membrane disappears. Nucleoli, those things that are making ribosomes, break down as well. They disappear. They stop producing ribosomes. And so the nuclear membrane, the nucleoli disappear, and the DNA appears. All of these happen during mitosis. Another thing that begins to happen at this point is the appearance of something called centrioles. Centrioles are little bodies found on opposite ends of the cell, little spots that appear on opposite ends of the cell. These will be places that all the components of the cell will move towards. Half of the components will move towards one centriole, half of the components will move towards another centriole. Actually, it turns out these are places where it's sort of like ropes are generated from these places, and ropes are sent out to all the pieces that are supposed to come there, and the ropes bring those pieces in, separating the components of the cell. So centrioles appear in this stage of prophase. The next stage, back when I was in school, the next stage was actually metaphase, which means middle phase. Now they've inserted another phase in between those, so it's pro-metaphase. This is a phase where spindle fibers grow from the centrioles towards the center of the cell. These are the ropes, the lassos, that are going to pick up things and pull them into the centrioles. We begin forming these ropes, which are made of protein, so they're protein spindle fibers that move outward from the centrioles, moving towards the center of the cell. When we move into the phase of the cell known as metaphase, middle phase, we find that the chromosomes that have appeared during prophase all line up along the equator or the middle of the cell. And think of this in three dimensions. You've got a spherical cell. So if you've got centrioles at, let's say, each end of the cell in this fashion, all of the chromosomes will line up in this circle in the middle of the cell. And the spindle fibers will grow from the centrioles all the way out to contact the various chromosomes in the middle. And specifically, the spindle fibers go from the centrioles to the centromere, that portion on the chromosome where the two chromosome arms are still connected, and that they come in contact at this point. So everything lines up in the middle of the cell at this point, all the chromosomes. And then we move into anaphase. Ana means separation. This is a separation phase. At this point, the chromosomes divide, break apart. Remember, we started by taking our DNA and duplicating it. We separated the two strands, made copies of the two strands, kept them connected at the centromere. And then now, at this stage, the anaphase, one of those copies gets pulled to one end of the cell, the other copy gets carried to the other end of the cell. The sister chromatids, that's the name we give to the two different strands of DNA, get pulled to opposite ends of the cell. And finally, we end up in telophase. Telo means purpose phase. This is what we're all about. When the DNA has finally been pulled to the centromeres, at this point, the DNA uncondenses. It now disappears because it's now going to go back into its long-stranded form. And around each centromere, around each end of the cell and all the DNA that's there, a nucleus will form at each end of the cell. And then the nucleoli will begin to reappear in each nucleus, and the centrioles, which no longer have a function of pulling everything in, will themselves disappear. At this point, we have divided the nucleus. We've created now 2 nuclei, one at each end of the cell, and the cell can now divide. This is the process of mitosis, dividing a nucleus to produce an exact copy of the original nucleus.